people who came. And I was going to do a tutorial on regression and just common problems that are cropping up in regressions. Because Sagiv said we can't have problems. I'll just talk about errors in regression instead of problems in regression. So we all do regression. We've seen 1 billion regressions during the course of this 2 weeks. We use them, in our GWAS. A bunch of people have been building networks or looking at correlations of gene expression. So it's kind of our bread and butter analysis tool for just about everything we've been hearing about, maybe with the exception of some of the assembly stuff. And when you took your class where you learned regression, they talked about all kinds of problems that can creep in that you need to be careful of. So things like having outliers, right, can bias your statistics. Or if you have something like population structure, you can have non-uniform p-value distribution under the null. So these are important things. They're just not the things I'm going to talk about. So these are really common errors in regression. But I want to talk about things that are still common, but maybe not so common that that everyone catches them all of the time. And in particular, a lot of these things are things that I'm seeing in lots and lots of published papers where I read them and I notice that there's some error, or potential error at least, that has snuck through and should have been at least asked about or talked about, and it happens a lot. And I've decided not to cite individual papers that are making these mistakes because they're so common. You can just kind of go to your last issue of Science or Nature or Cell or your favorite journal and find lots of these things. Not all of them, but a lot of them happening. So I'm going to talk about ignoring ascertainment, ignoring variance, and ignoring the induction of colliders when we're doing regressions. So first, ascertainment. So let's just start very simple. We'll imagine a prospective study on height. So we go out, we collect a bunch of individuals completely at random, and we measure their height. And let's just imagine we grab 10,000 people, and we look at some genotype, and we believe the genotype is the sum of 2 binomial random variables. So this is our model. And that height y is just 2 times this genotype plus some normally distributed noise. So we take the data and we make some histogram that looks really pretty, and we do compute this probability of y conditional on x. So we do prospective likelihood, just linear regression, and we see this slope, and we can compute a statistic over this, or we can measure the effect size. So what I mean by ascertainment is if we do something a little different, we go out in the world and we collect 2,000 of those 10,000 people. We look at heights of 10,000 people, but then we take 1,000 from one tail and 1,000 from the other tail. So we've decided to genotype just in the phenotypic extremes because genotyping is expensive. And now if we do this same sort of linear model, the same linear regression over these extremes of phenotype, we get this kind of slope here. So this is the original, the black points, the union of black and red points are all the original points, and this red line and the red points are the new slope you get from the ascertained study. And so we've done a good thing in the sense that we've increased power, but maybe we're unhappy about this new slope. Maybe we're more interested in the estimate of the parameter in the population than we were in determining whether or not the genotype was associated. So we have this sort of mixed bag of whether or not ascertainment was a good thing or a bad thing. So now let's move over to the case control world. So in case control phenotypes or dichotomous phenotypes, ascertainment's a lot more common. And so we'll think of the same kind of model. We'll have 10,000 people. We'll have a genotype, which is the sum of 2 binomial random variables. But now we're going to imagine that our phenotype is pushed through this logistic function. So now we're saying the probability of an individual being a case is some kind of logit over some affine term, and then as a function of the genotype as well. And now we're going to ascertain. So the reason it's so common to ascertain is because if you don't, especially for rare diseases, you'll need to do really tremendous studies. So if you have a 1% disease, a disease with prevalence 1%, and you want 1,000 cases, right, you need 100,000 individuals, controls. And so the trick has been to just sample cases and then sample controls. And so the question is, if we're applying this prospective logistic regression, but we're ignoring ascertainment, what is the effect on power, and what about effect size estimates? And so I'm going to, probably everyone's seen this, but just in case, this paper was kind of the fundamental paper that said why everyone loves logistic regression so much, or one of the primary reasons everyone loves logistic regression so much. And what it says is that whether or not I ascertain, I can do my prospective logistic likelihood model, and I can get the same effect size estimate. So that slope that changed in the case of height when I was doing linear regression, that doesn't happen to us through logistic regression. I get the same unbiased estimate of the odds ratio regardless of whether or not I do a prospective study and collect 100,000 people, or I collect 1,000 cases and 1,000 controls. So I can have a lot more power with unbiased estimates of my effect sizes. Okay, so now let's think about a slightly more complicated case. Now I have genotype and I have sex. So oftentimes when you read GWAS, you see people say, well, we conditioned on age and we conditioned on sex, and maybe we conditioned on BMI also. And maybe you do this to kind of remove some of the noise and try to increase your power. And so the question is, now that we've done this, let's think about the two different analyses we could have done. We can either look at this regression, the standard, sorry, I put this in kind of like R notation. I hope that's okay with everybody. If it's not, just tell me and I'll not write it on the chalkboard we don't have. Okay. So the question is, what are the effect size estimates? Does anyone have a guess? All right, and what about the standard errors? Well, if you don't have a guess for the first, you probably won't have a guess for the second. So the answer is, we're still cool. We still get unbiased estimates of the effect size, so that's really nice, because sex and genotype are independent. The problem is the standard error. So the standard error sorry, I have this backwards, actually. So this one has a higher standard error than this one. That is, I've lost power. So even though this Prentice and Pike result is telling me that I get an unbiased estimate of the effect size of G, it's not giving me any guarantees about the efficiency. And so I'm going to talk now about some potential solutions to this problem. In particular, I'm going to talk about retrospective versus prospective likelihoods. So what we've been doing when we're analyzing case control data has been employing this prospective likelihood with a logistic link function. So we have the probability of Y conditional on the genotype, assuming all the individuals are independent. This is the likelihood. And we just push it through this logic link function. And we're ignoring the fact that it was ascertained. And so really what we might want to consider is not the probability of Y, the probability of disease conditional on the genotype when we're doing retrospective study. We want to know what is the probability of the genotype conditional on the disease status. And so to do that, we're going to compute that, we're going to apply sort of two different steps that both came up in quizzes earlier this week. So let's see if anyone was paying attention. What did I use here to go from here to here? All right. I think you guys are just shy or tired or something. Huh? Bayes, yeah, I used Bayes. And this one from Saron's quiz? well, it's a law of total probability. So I can now write down a retrospective likelihood. So this is my standard logit link function. Here I'm going to pretend like I'm just going to use Hardy-Weinberg here. It's not 100% right, but it, you know, and we could fix that up, but we'll just pretend that it is and say probability of G is Hardy-Weinberg, and to get probability of Y, we're going to do this. And so in the case of a single covariate, when we're just looking at G, this has no effect, really. I can do prospective or retrospective, and there's very little that happens. But if we go back to this 2 covariate case where I have both genotype and sex, I can also think about a retrospective likelihood. And now things become a little more complicated. And to go from here to here, maybe I want to make some independence assumption. There's ways around this, but just in keeping with the very simple model, this is how we go from a prospective to a retrospective model. So I just have P of G times P of S over P of Y, and then here's my new denominator. And the kind of thing to notice that's a little bit weird, that you may not see, is that these aren't indexed by I. These come directly from where G is the, based on the allele frequency of G, and S is the probability of being male in the population, but they're not indexed by Y. So this is how you compute a retrospective likelihood. And if things are continuous, we just kind of change these sums to integrals. So we can compute a retrospective likelihood in most cases. And so let's take a look at one example where I set a true value of alpha 1 and alpha 2, and I consider a prospective versus retrospective likelihood, just like I wrote up. So we have genotype and sex again. So under the prospective and retrospective over 1,000 iterations, I get these unbiased estimates. And now I'm going to look at the improvement in efficiency in using the prospective versus retrospective case. And the way I'm going to think about efficiency here, there's lots of different ways to think about efficiency, but I'm going to think about the ratio of the standard error around my estimates, since that's, for example, what's driving the power. If we think about a Wald test, it's sort of the relationship between the effect size estimate and the standard error. And so when this number is larger than 1, it's saying that the standard error of the prospective is much larger than the standard error of the retrospective. And so this is agnostic, meaning that I don't have any external information. So I just apply that retrospective likelihood as I showed it on the last slide, and I sort of sum over, as I'm fitting my effect size beta, I'm computing this likelihood. I'm trying to maximize it conditional on the parameters for these alpha 1 and alpha 2. And what you can see is that for alpha 1, at least, it was much more efficient to do a retrospective likelihood. In the case of alpha 2, it was much less efficient. And so sometimes it can be good, sometimes it can be bad. From a computational perspective, it's bad. It's very expensive because we have to do this. Every time we choose a new value of alpha 1 and alpha 2, we have to plug through this whole machinery again. And so doing the optimization is much more expensive. In the literature, and I put a bunch down here that I'll kind of walk through in a second, one of the popular ways to use retrospective likelihoods is to incorporate external information. So I might know things like the disease prevalence, or I might have some estimate of the overall frequency of the genotype in the population, or I might know what proportion of the population is male. And so if I take the known frequency of sex and I plug it in, instead of just taking the estimate that's coming out from my population sample, I get a much bigger gain in efficiency using the retrospective compared to the prospective. And if I know both PS and PG, you have a really massive gain in power that's available using the retrospective versus prospective. So when we're doing these ascertained case control studies and you want to think about using covariates, you can be shooting yourself in the foot if you're putting them in there for the purposes of trying to improve power. There's other reasons to use covariates related to, correcting for confounding in structure, but when you include something like sex and you're doing a study of autosomes, the purpose is not to correct for structure, it's to try and improve your power. So when you see someone doing a study of type 1 diabetes and adjusting for sex and not doing a retrospective likelihood, at some point they might be sort of throwing away a bunch of signal that they could have recovered had they done a retrospective analysis, and at least over the signals that are sort of in the top end of the tail, if you're worried about computational speed. So there's been a lot of sort of work on this. This is a method, one of the first ones considering retrospective likelihoods in genetics, that used a semi-parametric approach to kind of speed up that integral and allow for some correlations between genotype and environment. We wrote a paper a while ago trying to use data from CDC as the external source of information to increase power. And then there's a bunch of work in mixed models. So you can imagine when you're doing a mixed model analysis, because we have something like, say, UK Biobank, where there's a lot of structure, and we have some kind of ascertainment going on, we can also run into problems. And in the case of mixed models, we're not just trying to compute association statistics, we're also trying to do things like risk prediction and parameter estimation of things like heritability. And ascertainment can bias heritability estimates and can reduce predictive power. So some of these papers sort of discuss ways to go about using retrospective likelihoods, or at least addressing the ascertainment in case control data where we're trying to employ an LMM. I didn't want to go through it in this talk because it's kind of involved, but I thought I'd leave the citations and just mention it as a problem that you need to think about when you're using linear mixed models and ascertain data. The thing so none of these are except for the heritability analysis stuff, none of these are causing false positives. But another type of analysis that gets done in ascertained data very often is something called secondary phenotype analysis. So if you're interested in some disease like coronary artery disease or schizophrenia or autism or anything that is at a rare frequency, you might want to study the tissues involved, for example, in those diseases. So you might say, I'm interested in coronary artery disease, so I want to look at hearts of people who have had a heart attack. So you're ascertaining on disease status, and then you're looking at features of the heart, for example, gene expression. And so there's a lot of papers floating around that look at these co-expression networks or gene networks or clusters or whatever it is, metabolic networks or metabolic flux inside of tissues that were on ascertained data. And here, it's not necessarily a question of efficiency. And to show that, I'm going to walk through an example. This is also kind of a twisted example of Simpsons paradox, if you're familiar with that. So let's consider two variables, x1 and x2, that are normally distributed. And we'll make a model for y, which is x1 beta 1, x2 beta 2 plus epsilon. So if we plot x1 against x2, we get this nice round, multivariate normal with no covariant structure whatsoever, right? The correlation is 0, it's a perfect sphere, disk. Now we're going to ascertain based on y. So we're going to say we want people at the extreme tail of y, people who've had a heart attack, people who have autism, and we're going to look at x and y again. And these are the red dots. And so what you can see is this anti-correlation between x1 and x2. And so the point is, if you have things that are marginally associated only in the population, they're just increasing your risk for getting a heart attack, and then you do RNA-seq in hearts, you're going to see all kinds of trans structure, correlation structures. And so if you see papers that are talking about this, they may very well be full of statistical artifacts. And I think nobody's really accounting for this right now, and I think it's an interesting problem to be thinking about. So just as like a summary for this section on ascertainment, ascertainment is an effective way in terms of study design to increase our power, right? We can get a lot of cases compared to a prospective study, especially for a rare disease. The effect size estimates from a linear regression are perturbed by ascertainment. So you can't do a study of effect size heterogeneity between one study that ascertained and one study that didn't when we do linear regression. And in particular, if you used a linear mixed model and you just got a beta, linear mixed models are not logistic models. They give you some slope. And so if you ran an LMM to do your GWAS and you get some effect size estimate and you don't apply some kind of transformation to shift it to an odds ratio, you can't compare two different studies and expect there not to be heterogeneity if the sampling scheme was different. Logistic regression in many cases provides unbiased effect size estimates even with ascertainment. So this is the Prentice and Pike paper. But conditioning on covariates, including, that's one too many M's, can, yeah, well, maybe we have now the multi-blup thing's very popular. Multi-mix models. All right, so retrospective likelihoods can recover this power, but it's computationally expensive. And so, here's sort of an open problem people might want to think about is how to do these kinds of things efficiently. And then maybe most importantly, and also most unexplored, is that ascertainment can induce correlation of secondary phenotypes that influence disease risk. As a side note, ascertainment also induces correlation of secondary phenotypes across cases and controls. So if heart attacks cause a perturbation in the expression network, and I look at correlation across cases and controls, then I have different means in the hearts of people who've had a heart attack than in people who haven't for the things that a heart attack change. Or if you're looking at microbiome, it might be the case that microbes in people who have some gut disease are affected by the gut disease, not the other way around. But if I look at PCA or I look at correlation of pairs of things across both cases and controls, or if there's some kind of subphenotype heterogeneity, for example, there's different types of schizophrenia. One has genes affected, genes A and B affected, and the other does not have genes A and B infected. These types of causal structures from the disease to the phenotypes can also induce correlation. Okay, so problems everywhere. In the next section, yeah. 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 Well, I mean, if you don't rescale, so if someone did like a study of coronary artery disease and they had 1,000 cases and 5,000 controls, and somebody else did and they used a linear mixed model, and then as a phenotype, as an outcome. No, that's what I'm saying. If you did a dichotomous phenotype, and you, but people are, well, you do and you don't. People, I mean, it's sort of the standard. Okay, you could use a logistic link function. So you can do a GLMM, but you don't have the same guarantee as the Prentice and Pike result when you're doing a GLMM when you run it through an ascertained study versus not ascertained. So even if you pay all of the cost of doing a logit link function in your LMM, you're not promised to get in the same odds ratio in an ascertained study versus an unascertained study. So even there, it's not like whether you treat it continuous or you don't. There's some literature on how to go from a linear estimate of effect size, treating 0 and 1 as continuous, back to an odds ratio if you know the prevalence of the disease and you know the number of cases and controls. And so, it makes some assumptions, but one.
That was, I see, because then it combines the studies by distribution.
Yeah, so as long as the assumptions of those, so it's D and L, do you remember who wrote about this stuff? I'll get it to you. I think that it's, I'm not going to say it because I'll get it wrong, but there's some literature on how to take a continuous estimate and back out an odds ratio and a standard error of that odds ratio. But I'd say it's pretty rarely done. Okay, so the second thing I'm going to talk about is ignoring variance. For a lot of phenotypes, variance and mean are correlated for pairs of phenotypes. So here's some data we have on FEV. It's a measure of lung function, and here is age. And so you can see that as you get older, while you grow, your lungs get bigger. But the other thing you can see is that, the error is also increasing with age. And so if we think about a linear regression of FEV on age, the errors don't have an age term in here, so it completely ignores this. And so, when you don't have a model that fits the data well, one of the costs is power. So if we somehow got age into this epsilon term here, we might have more power to recover this relationship between age and phenotype. But our question is about errors, and so we want to know, and I guess not having optimal power is an error, but I want to talk about a different type of error, is false positives. So I want to know, are there cases where ignoring mean variance relationships can induce bias? And so I'm going to consider the case of adjusting for structure and association studies. So let's think about like an admixed population. So we have population 1 and population 2, and we make our cases and controls, and we can have, so within population 1, the allele, you have more of these AA, and in population 2, the little a is more frequent. And we consider linear regression, and we evaluate this sort of lambda GC, which is a measure of inflation, when we have an admixed population and we just run linear regression. And you see this kind of lambda GC that's inflated. So this is a QQ plot. And when this value, the median test statistic, is greater than the sort of expected median under 1 degree of freedom, we say that there is inflation. And there's inflation if, for example, individuals from this population were more often cases than individuals from this population, then any allele that's got a different frequency in this population will be associated, and so we get inflation. And so we need to run linear regression with PC, so this is why everyone is busy doing this, and we get a much nicer distribution of test statistics. And so the warning here is that the assumption is that linear regression with PCs assumes that ancestry, or that the PCs only affect the mean of the distribution and not the variance of the distribution. And so we want to consider the possibility that variance changes with ancestry proportion or with the PC. And so the first thing we're sort of asking is, we want to know whether, when you have ancestry correlated with a phenotype due to genetic reasons, we should expect a relationship between PCs and variance. And so we simulate G, we simulate effect sizes, but in order to induce a relationship between ancestry fraction and phenotype Y, we need to make the betas correlated with global ancestry fraction. So we have sort of 3 different strategies for choosing betas. One that's completely random, so this is going to have no population structure. One odds and fixed, and these are just different ways to induce a genetic basis for correlations between theta and y. And when we simulate data like that, where we don't simulate it randomly, but we induce a correlation between theta and y due to genetics, what we can see is that when theta increases, the mean increases, because that's how we've sort of chosen our betas. But this also induces a relationship between theta and sigma squared. So to the extent that selection has induce differences that in phenotype, it's quite difficult to have theta be associated or PCs be associated with an outcome without having a concomitant relationship between the PC and variance. And so what is the consequence of this? Well, if we simulate data with no genetic effect, and we test for, we do basically a GWAS with PCs, and we consider 3 types of SNPs. We consider one SNP that has a minor allele frequency of 0.5 in population 1 and 0.05 in population 2. And what you can see is that even though we put in PCs, we have a lot of inflation. When the minor allele frequencies are equal, we have no inflation. And when the frequencies are going the opposite direction, we have deflation. And what's driving this is that the variance in population 1 is going to be very different than the variance in population 2. And because ancestry is now tied to variance, sorry, I should have said that here, our epsilons now are just are 0, but we have an influence of ancestry on epsilon. And so when we generate data under this model, we have these sort of strange QQ plots where there's a lot of inflation and deflation. And this also includes biased effect size estimates. So if we actually had causal variance, the estimates of the effect size of the causal variance with linear regression with PCs are incorrect. And the kind of sinister fact of this problem is that the tool that we most commonly use to diagnose inflation in GWAS is this QQ plot, and we never split our SNPs by minor allele frequency in the different populations when making these QQ plots until we saw it first in this paper. It was a really clever idea to do this, and the problem is when you sum all of these points together, you get this beautiful QQ plot. So there may be QQ plots that are floating around that you look at and you say, oh, this is wonderful, but in fact there's a bunch of inflation and deflation of individual test statistics that are living under the hood. And so the question is, how do you model variance when you're doing your regression? And there's a lot of different approaches, including some really cool work from Barbara, who you'll hear from later, and she also thinks a lot about variance, but I think less so for population structure and more so as just an interesting phenotype in and of itself to look for relationships between genotype and variance. So here's sort of the, a lot of the literature. We chose this just because I knew about it, and it's simple to think about. It's something called the double generalized linear model, and we call this one the ancestry DGLM, which is just a DGLM where we allow ancestry to affect variance, and I'll show you what that looks like. So so we have Y, our phenotype, G, our genotype, theta is our global ancestry, and we have this term sigma theta, which is the ancestry variance component. And when we do linear regression with PCs, we have this null and alternate that look like this, where epsilon is just n0 sigma squared distributed. So standard linear regression. When we run a double generalized linear model, it looks exactly the same, except epsilon is different. And epsilon can be different in lots of ways. DGLM is sort of like, well, it's generalized. So that, just like in generalized linear regression, you can have lots of different link functions. Here we can have a multiplicative effect or an exponential effect or logistic effect or whatever is right. We can use non-normal distributions. We can use a negative binomial distribution or a Poisson or whatever the appropriate distribution is. And we don't have to use ancestry here. Like in that case of age and FEV, we can put age in here and try to improve power by allowing age. And so there's a couple trade-offs. One trade-off is that this is more computationally expensive to run than linear regression. We can't just do our X transpose inverse, X transpose Y trick and get our betas out. You have to do some fitting. But the benefit is that, well, I'll show you in a second. So let's just look at some simulations. We'll have population 1 and population 2, and we'll look at 100 individuals, and we'll just choose one SNP, and we'll have in population 1, we'll have theta equals 1. In population 2, theta will be 0. They'll have different maps. And so this one has a different variance than this one. Population 1 has a higher variance than population 2. Okay? And this is our model for y, g beta g plus theta beta theta plus epsilon, where this is our distribution of epsilon. All right, so if we think about the double null, and we look at ancestry DLM and linear regression with PCs, this is the power, so it's 5%, and so everything's kind of cool under the null. The caveat that I forgot to make before is that in addition to having a higher computational cost, you have to think about outliers differently than you were thinking about them. You know, outliers are always a problem, right? We can't have outliers in our genotype space, right? We tend to filter things with a super low minor allele frequency because they're rare, or you know, we do some kind of normalization to gene expression, both in our random variables and in our outcomes. It happens to be that most of the link functions that we choose in DGLMs are more sensitive to outliers than they are in linear regression, and so just keep that in mind if you decide to play with these tools. So here's this other case, which is that when you have no sigma theta, that is the variances are the same, the power is similar. So we're not losing much by using this ADGLM. And if we look at the null, well, we get back to this problem where the linear regression with PCs for two different kinds of SNPs, unless they're exactly equal, have power that's greater than 5%. So we have inflation with linear regression with PCs, and we have a good distribution with an ancestry DGLM. And so if we go back to our QQ plots, we can redo those simulations now comparing linear regression with PCs in the red to ADGLM in the blue, and you can see across all three of these things, we get this really nice null distribution. If we now look at power, where we have sigma theta not equal to 0 and beta g actually has an effect, the power here is slightly lower than the power of linear regression with PCs, but in the case where these are equal, by having a model that closely fits the data, we increase power, and here we get a much larger gain in power because the variance here is higher, and that's what we're modeling. And so if we redo those three QQ plots with the same three minor allele frequencies, P1 greater than P2, P1 equal P2, and P1 less than P2, you see that in that case where P1 is greater than P2, ADGLM has a much higher power than linear regression. It has slightly higher power when the allele frequencies are equal, and when the allele frequency is much lower in population 2 than population 1, ADGLM loses a little bit, but when you integrate over all three of these things, you end up winning. So this is basically a table showing the same thing. So that's all I'm going to say about DGLMs, other than there's great packages that are available in Python and R. They're very simple to use, and so I think there's no reason not to play with them if you're if you are observing in your variables that there's relationships between mean and variance, or if you're doing GWAS in an admixed population, I think these will be very nice, including you can push the whole DGLM framework through a probit model, and so you can still look at a case control phenotype and have a DGLM. Okay, the last is everyone else went over, right? So I'll go over to, I want to talk about collider induction. Now when you write a title, these are newer slides, when you put a title into PowerPoint in like 2016, it gives you like all these awesome options. I didn't do this like blue circles thing, it just was proposed to me. It's really cool. All right, so basic severial adjustment, we have some Y's or outcomes, some genotype that we're testing, and then some covariates that also influence Y. And if we think about these things in terms of variance, we'll just say C1 is completely made of C1, C2 is completely made of C2, and Y is a little bit of C1, a little bit of C2, a little bit of G, and then a bunch of other stuff that's independent. And so if we, these slides are from Hughes Achard, by the way. If we condition on, that's weird. Oh, okay, Andy and Hugh are doing a lot of this work. If we condition on C, we've increased the proportion of blue relative to the amount of red, relative to the amount of blue that's left, and that's why conditioning on things like sex and age can increase the power in our GWAS, because we threw out age and sex, and so the haystack got smaller and the needle stayed the same size. Okay, so in a more realistic case, we don't have like the things that are directly impacting Y, but we have this thing C that's made up of C1, C2, and some brown stuff that's independent. And now if we adjust, right, we fit the model alpha C, and we're subtracting that out of Y, and so we remove big chunks of the green, but we add a little bit of this brown in. That's what's going to happen when we adjust Y, because we're taking Y minus alpha C. And so it's not as good, but still overall it was a win to do our adjustment. So the case to watch out for that's often not watched out for, and it's crazy when I see, I've collaborated now with some colleagues who are now more on the like epi or medical side of things that aren't looking at genotypes, and they get these review comments back from their reviewers that are like, you should add in 5 more covariates, like time of admission to the hospital, SES, and blah, blah, blah. All right, so let's see why just throwing a bunch of covariates in for no reason can often be a really bad idea. So now we change our causal graph, right? And for those of you who, well, I guess if you know a bunch of causal stuff, you know what collider bias is already, but this is the case of collider bias when we condition on C instead of just looking at the relationship between Y and G in the regression. Because what happens is when we condition on C, now because C has a little bit of red in it, the red goes in. And so when we're looking at the relationship between the genotype and Y adjusted for C, there's going to be a correlation. So we've induced a relationship between G and Y by conditioning on C. That is, we've created a collider, and so we get a false positive. You know, there's lots of other graphs we can draw, and I'm not going to draw them because we're short on time, but this is the thing to watch out for. And there's this new paper that's up on the bioarchive that was sort of looking at this in response to a paper Hugo wrote about sort of collider bias induction, just to see if something like this could actually happen. So if you take UK Biobank and you do an association between sex and the autosomes, that is, sex is my outcome, and I'm only looking at the autosomes, right? So there should be absolutely no signal. And in fact, if you just do that analysis, sex is the outcome, and I do a GWAS for sex on the autosomes, nothing shows up. But if I adjust for height, you get a bunch of signals back at the SNPs that are associated with height. So okay, so what does it mean? It means that if you're adjusting for height and some other GWAS that you're busy doing, and there's a relationship. So in this case, there's a difference in height between males and females, and that's why this is sort of going on. But if you're adjusting for height in some other study, and there's a relationship between height and genotype and height and outcome and all of this noise, you can make a bunch of height SNPs show up in your GWAS results. And so I don't have like a nice solution about choosing covariates like I had with the GGLM and the retrospective likelihoods. So in this case, the kind of story is just be careful and think about what covariates you're throwing in. Ask yourself why am I including this covariate other than like my PI told me I should put this in or the reviewer told me. You can tell the reviewer, no, that's a bad idea, you know. Okay, and then just to end up, I want to talk about some work that Andy's been doing, which is thinking about the use of PCs or SVs or peer factors when you're doing gene expression analyses. So in these studies, you make these new covariates, and they're not there necessarily for the purpose of correcting for structure. You're worried about, you know, batch effects and all of this noise that is living in your gene expression data. So if you're doing an EQTL study, you're asking about gene expression that varies between the exposed and unexposed cells in some molecular experiment. You will oftentimes run one of these packages and then ask about relationships between variables conditional on these peer factors. So I want to know, is this gene have a different mean in expression when I give it this drug versus when I don't, conditional on the PCs of, you know, or the peer factors or the SVs. And so the problem is if we have some effect here, so here are Y are the gene expression values, and U is this PC, and so all of the Y, right, the PCs are just linear combinations of the Ys, and if we have this effect of G on one of them, and we look at the effect between G and say Y2 or any of the other ones conditional on U1, for the reasons we talked about before and other reasons that Andy talks about in his paper, you can induce a correlation that wasn't there before, and there are supervised versions of these methods, and in many cases they work well in practice, but they can sometimes not work well in practice, and kind of messing around with some stuff in the past couple days showed that you can break these things. So you can come up with a simulation where I supervise one of these things and tell it not to make things independent of that G or independent of age or independent of drug, so that I can I can make these structures and include my PCs to improve power, but they won't. And so I think this is a really cool problem that may or may not have a solution, which is, you know, can someone come up with a way to do dimensionality reduction conditional or with knowledge of the variable that I want to regress on, like genotype or exposure, and make those PCs independent of that while still capturing structure effectively. And so that was one bit of the summary, and the other is just what I said before, that conditioning on covariates, including PCs, can induce these colliders and create bias. So yeah, thank you. Questions? Yeah.
Could you say something about principal components derived from genotypes and how that could possibly induce collider bias? Not using like linear mixed models, but just using a linear model or linear regression.
Oh, that's interesting. So I guess it would have to be about LD, right? Okay, so if you have an admixed or if you have a structured population, then you can have genotypes that are that are far apart, that are correlated, and they might be captured in a PC. It's hard to see the causal graph, but I guess the idea would be if you have some genotype over here that is influencing the trait, you have some genotype over here that's correlated to the PC only through the fact of admixture, then maybe you'll see an association between this genotype and the outcome. But I'm not sure that. I think you might be safe because I think you need some environment that's influencing, and I'm not sure if genotype counts. Let's talk about it. Yeah. Yeah.
If I have some covariate, but I'm not sure that you include them all. So the safer run like one with the covariate and the other time without the covariate, and in both cases I get significant results. Would you say that then I'm on the safe side?
It depends on whether or not the purpose of including the covariate was to improve your power. So, if or to account for confounding.
Yeah.
So, if the purpose is to account for confounding, well, if you know that it's only correcting for confounding, then I guess removing it's not interesting. Where you can get into trouble, the only way that it's a good idea, the place where you could get into trouble is if you had a variable that was both correcting for confounding and inducing a bias. So when you remove it, it's not perfect, and it seems quite unlikely. So I'd say overall it's a good idea, but you can write down a model for a phenotype in which removing it will cause a false positive, and including it will cause a false positive. If you want to be really evil, you can make such a thing show up. There's a paper on the archive from Hughes Achard that has from Hugo that is proposing another approach for selecting covariates that I think is interesting. I'm on the paper, so I'm biased, but I think it's pretty interesting. And so the idea is when you're trying to, what you really want when you include a covariate that you're including for power is you want it to be independent of the genotype or the variable that you're testing. So if you have G and C here, the cases where everything was cool was when G and C are independent. And basically all of the cases that are bad are when there's a relationship between G and C, either like this or when G also has an arrow to Y. And so the proposal in that paper, so normally you reject, you can do this hypothesis test to ask whether G and C are correlated, and you can look at the sort of null distribution like we always do and ask whether the test statistic falls outside of that null. And in his paper he makes this proposal to look at both the standard null as well as the conditional null on Y. And so you get these kind of two different null distributions, and you ask whether the relationship between G and C lies in either one of those, and you do a better job of filtering covariates with that approach. All right. Any more? All right, thank you.